Well, hi again, Mr. Bleeker here. We'll finish up our last section on the structure of DNA by looking at how it's organized. I'll just pop over here for a moment before we start formally. Now, you're looking at the nucleus of a cell, and a lot of people think that our DNA is kept in these packages, right, called chromosomes. But that's not what happens all the time. In fact, for a majority of the cell's life, when it isn't dividing, when it's growing and becoming bigger in something we call interphase, chromatin, see these dark, sort of spindly fibers here, that represents the threads of DNA that are massed in the nucleus. This one in the middle we'll talk about a little bit more. That's called the nucleolus. Spoiler alert, that's where ribosomes come from. But for now, I want you to think of sort of snot-like threads of DNA that are loosely aggregated in the nucleoplasm, aka the cytoplasm of the nucleus. So let's get going. So we're going to finish off on our last lecture with it's the same slideshow with the history of structured DNA. So I will get reflector running and we'll get right to it. Okay, here we go. So when last we met, we were talking about the semi-conservative replication of DNA. That was on about slide 20. Just pop down here a little bit, and there's just this tidbit left, which is all about DNA organization. Now, DNA is arranged into chromosomes for the purpose of cell division. So let's make a note there. And the reason that's important, think about it this way. If you've ever moved, you put all your belongings into boxes and then you move them to your new home and you have them well labeled and you know where everything is. That's really important from a cell biology standpoint. When DNA is replicated, you want to have all the genes and all the DNA. Humans have 23 pairs of chromosomes, so you can think of it as 23 large moving boxes that contain all of your genes. But if you're not moving, you don't need the boxes. So generally, DNA in loose form will be in a form called chromatin. Now would be a good time to show you this one picture, and I think it just clarifies it all so nicely. There we go. So we'll look into the nucleus here just specifically. And if you look at the chromatin fibers, now there's distinct territories, but that's not the point right now. We'll look at some of these threads, and if you look closely enough, there we go, you'll see this almost like floral arrangement. And those are the chromatin threads, and they're looping and looping and looping. So we talk about this as the tertiary structure, the globs. And if you start focusing in on that and you look in there, you'll see these repeating sort of sequences, and you see those little green balls. Like, what's that? Well, the primary structure, here's your DNA, your twisty ladder, and that twisted ladder can be woven around these green masses. They're little proteins that we call histones. And as we wind the DNA around them, what we get is primary chromatin structure. It's very small, 10 nanometers, looking at here in its overall scope and size. And as we wind it around these, this gives it enough structure. And then we begin to wind the histones that are bound with the DNA around and around and around and around. That gives us our next structure, our secondary structure. And if this is then wound around and around and around we go, you get to sort of larger masses. So your DNA is basically wound around these little spools that we call histones. Now here's a neat little animation. This time we'll look at a chromosome. It's the same idea. I just thought it would be neat to show you. So we'll zoom in, and you can see the same kind of coiling aspect. And as we pop in a little bit further, you can see now in blue the DNA wrapped around the histones. And if we go in a little bit further, there we go. Now we can see the twisted ladder, as if it's contained in sort of like an organizer here. Just easier to see it that way. And at this point, when we've got DNA wrapped around histones, what we have is what we call a nucleosome. It's just a stabilizing structure. So there you go. Reset the animation. Okay. Pop out and just finish off our note material here. This is just one of the shorter topics for discussion. Here you go. So you can see your nucleosome. And whether or not DNA is floating around as chromatin, or whether or not DNA is a bit more aggregated into chromosomes, you're still looking at fundamental primary structures here, which we call nucleosomes. We just build these up until we get sort of large superthread globs. And ladies and gentlemen, that is it. That's how DNA is organized in your cells. Well, more specifically, how it's organized in your nucleus. Don't worry, we'll talk about the nucleolus soon enough. Okay, ladies and gentlemen, thanks for listening. Short, sweet. Have a good one.